This is Cheloniac episode 44. Today we are joined by Dr. Arthur Georges, who is a distinguished professor at the University of Canberra. Dr. Georges is the former foundation director at the Institute for Applied Ecology at University of Canberra. He's also on the steering committee for the IUCN Tortoise and Freshwater Turtle Specialist Group. He's done an incredible amount of work looking at the ecology, evolution, and systematics of turtles and tortoises. Focusing on Australian Chelids and Australian turtles. We're really excited to talk to him today. Thank you for coming on.
Yeah, great to be with you.
So, Dr. Georges, what first got you interested in turtles?
I guess, it's a funny thing that many people sort of work out what they're going to do by age 7. Like, I don't know if you've seen that 7 Up series where they follow people every 7 years starting at age 7. And quite a few of them have said what they wanted to be when they were 7, and it transpired that that's what they became. And so I had some turtles when I was 7. I was very proud of it. I had more turtles than anyone else in the suburb, and I had a father who was tolerant enough to build me a pond in the backyard. And I learned a lot about turtles as a youngster, and I guess that might have set my trajectory sort of without me knowing. To be fascinated by turtles and to move into that area of research.
That's cool. It's a lot of people. For us, I think it was very similar, and we're sort of younger still, but kind of getting involved early on, and have just carried along with the passion.
I think that's one of the few stories we've heard so far that doesn't directly involve red-eared sliders.
I didn't see red-eared sliders, but I had the eastern long-necked turtle, which was fairly common at the time around Brisbane. Not so much now. I think maybe knocked out by the cane toads. And my cousin had he collected turtles from all over the state, and so that sort of was pretty interesting to me. He was a lot older than me.
Maybe we can start with that, actually. That was something that your lab's doing a lot of work with, the eastern longneck and tracking movement and sort of response to urbanization. Maybe just you could give us a rundown about what you found that's particularly interesting with that species and sort of how they're distributed.
Yeah, so the eastern longneck turtle's really widespread in eastern and southeastern Australia. And it's got a really interesting life history, sort of a cryptic life history. It spends a lot of time on land. And I mean, that's not unusual for some of your North American species, but in Australia with Chelid turtles, that's quite unusual. So these animals are ephemeral water specialists. So they really do well when the periods are wet and the swamps and rain pools and across the landscape. And they get out into that area and they get out there and there's no fish there because fish can't get there. No competitors. They eat earthworms and all sorts of food that's in the productivity boom following the flooding of wetlands, and that's their specialisation. But of course, in Australia, we're a land of flooding rains and droughts, and when the droughts come, all those areas dry up, and so they make a choice. They either go up into the terrestrial areas and excavate underneath the leaf litter, and they can last up. But we've monitored them for over a year in a terrestrial situation without going back to water, or they migrate across land to permanent water that's within their home range. And so they've got these dual strategies, which is really interesting. And we've also looked at how they navigate, and they use the sun to navigate. So what happens when it rains, the turtles all come out of the permanent water, and then they sit and wait until the cloud goes away. And once the sun comes out, they navigate out to their ephemeral water bodies where they really boom. So they've got this sort of pulse that goes on. Our drought cycle could be from 7 to 15 years. They have this pulse going where they move out into the ephemeral waters, come back to the permanent waters. When they're in the permanent waters, incredible densities. They stop growing, they stop reproducing, they just sit there and wait for the 7 years or whatever until the drought breaks, and then out they go again into the ephemeral waters, and then they grow and breed, and everything happens in those sort of ephemeral swamps and wetlands. So really interesting life history, I think.
One thing about them that was kind of stuck out to me, I've done some work with looking at the impacts of urbanization on pond turtles in California, and you've done some work that the eastern longnecks seem to come into contact with people a lot in high urban centers.
Yeah, so we have a lot of we build water retention ponds when we build our urban environments to stop the sedimentation getting into the rivers. And the turtles really do well in those urban sort of retention ponds. So they're really large lakes inside the city bounds. And the turtles do really well in there. And they do still do this cycle of moving out and back in. But the difference is that they tend to move straight from the permanent water to the swamps and back again in the urban context, whereas in a natural context, they spend a lot more time sort of wandering around in the terrestrial environment, burying under leaf litter, etc. Well, in an urban environment, they don't do that. They tend to
I don't know
just
if you can still
high foot it, to the swamp. Whoa! Something happened then.
Yeah, did it disconnect? I think it's it may be on my end. I'll fix it. 1 second. Sorry.
Well, it was going so well.
We'll start that one again.
Yeah, sorry about that. This is me. I've got to connect to a different Wi-Fi.
Yeah. Well, I live in the country, so I'm using Starlink.
Your connection is like surprisingly good now, so better than most of our other guests we've had on.
Okay.
Hello?
Hello?
Hello.
Can you? All right. It sounds good. You're frozen for me. Everyone's frozen for me, but it's definitely my end. 1 second.
He's not frozen for us, though.
I can see
him.
You guys are good? Yeah.
Good to see you've got pants on there.
Okay, there we go. We should be good now.
All right, ask that question again then.
All right. It was good up until talking about. Yeah. Well, okay. We'll just restart the whole question. That works. Okay. So in terms of something that kind of stands out to me with the eastern longnecks, it's interesting is they seem to inhabit or come into contact with people fairly frequently in urban centers and can even sort of thrive in those areas. You've done some work with that. I'm curious what you found there.
Yeah, so we have a lot of urban wetlands that are built as retention ponds when we build our suburbs so that the sediment doesn't get into the rivers. And those retention ponds are like urban lakes, and the turtles love those urban lakes. So they get in there and they do really well. They still undergo that sort of cycle of moving out into ephemeral waters, but in the suburbs, they tend to move directly from the permanent water out into the swamp and then back again. Whereas in a natural situation, they spend a lot more time on land, sort of estivating under leaf litter and in logs and various other things that are unavailable to them in the urban environment. So their behaviour is adapted, but they do really well in that situation. The other thing is that in the urban situation, they come into contact with cars. And it's a terrible thing, the number that get killed on the roads in Australia. So when it rains and then the sun comes out, they all move. And they're moving across the roads, and of course they get killed on the roads. So there's a lot of effort going into providing underpasses and various other avenues for the turtles to move through the urban environment, at least in Canberra, whereby they don't suffer that terrible mortality. So that's an issue, the road mortality.
Right, it's interesting. I've seen a similar kind of trend with pond turtles in California and road mortality being an issue. Some of the other, this is kind of always the question I think a lot of ecologists get with turtles is what is the worst stressor that they're facing? And that's it can be a hard question to answer. With respect to the eastern long-necked turtle in particular, is there a threat, or is road mortality the worst threat sort of facing that turtle, or is there something else, or is it even possible to rank the worst situations for those turtles?
Well, we just wrote an article on this for Australian Ecology. It'll be out by October, I think. And in there, we covered the threats against all the species in Australia. And the eastern long-necked turtle, I guess the biggest threat for it is the loss of the ephemeral wetlands. So Australia is the driest vegetated continent on Earth. So the total sum of water flowing out of our rivers is less than flows from the Missouri into the Mississippi. And so that's one difference about Australia compared, say, with North America. The other thing is that the climate's really unpredictable. And so you have these long droughts, you have incredible unpredictability, and these animals are moving and adapted to that unpredictability. They're moving out in ephemeral wetlands. And of course, we're changing the flows in our rivers so that the floodplains don't flood as often, or they flood at the wrong time. And so there's a massive loss of opportunity for these turtles. So I guess the habitat loss and the draining and alteration of environmental flows is the biggest threat to this species in terms of the reduction of numbers over time. Now, I'm not saying they're not abundant. They're still abundant, and there are counterbalancing influences. So the proliferation of farm dams across the landscape has benefited this species. So there are sort of counterbalancing influences, but they're nowhere near as abundant as they used to be, where hundreds and hundreds of them used to cross the Princess Highway every time it sort of rained, and often seasonally rather than once every few years. And that no longer happens. I mean, they don't cross the Princess Highway in hundreds anymore. So their numbers have declined dramatically from what they used to be, but they're still quite abundant within their range.
Right. It's some level of conservation is trying to keep the common species common. But yeah, it's interesting to quantify what are the biggest threats towards that species to try to devote efforts to the thing that's most necessary, right? That's kind of the goal.
Yeah.
And something else that sort of has plagued that turtle and potentially some other species is salinization of water bodies due to altered hydrology and such. Maybe you could talk a little to that and what causes that and what are the issues for turtles?
So salinization is a big problem in Australia because there's a large amount of salt in the below the surface in Australia, in particular areas that were once inland seas. So there's a large amount of salt, and our land use practices and the changes in land use practice have often drawn that salt to the surface. And that causes a lot of problems for agriculture, but it also causes massive problems for the freshwater biota. Now, the eastern long-necked turtle, I guess, is perhaps more adapted than other species to salinisation. They do live in brackish waters, and so basically they're more tolerant than a lot of other species. The other snake-necked turtle in the Murray-Darling, it is not as tolerant to salt water, but it has all these behaviours. So when it rains, the fresh water sits on top of the salt water, and they drink that, and then they don't drink when they're in the brackish water. So they've got all these different ways of adapting to salinity, which is a feature of our landscape. One of the things that happened with the long-necked turtle down to Lake Alexandra at the mouth of the Murray is that the habitat became very brackish, and marine species moved into that area, and a tube worm in particular started to colonise the turtle's shells and built up an incredible crust on the turtle's shells to the point where there are just little holes where the feet came out and a little hole where the head comes out, and the turtles could hardly move. So that would obviously cause mortality. It's sort of an unusual situation where the salinisation of their habitat brought in another species, which then encrusted the turtle's shells and led to mortality. So it's sort of, a bit of a surprise, that one.
It's interesting that it was so much of a sort of influence that it led to the ecosystem change to some respect.
Yeah.
It's almost, it reminds sort of, it's a different situation. This isn't natural, and it seems like it's exacerbated by agriculture and sort of leaching of salt into different systems. But kind of the diamondback terrapins in the United States naturally occur in salt water. But it seems like the long-necked turtles, chelids, are fairly adaptable. I mean, what level of salinity are we talking about? And is anything known about how they sort of control blood osmolality, that sort of thing?
Yeah, so a former student of mine, Deborah Bauer, who's now at University of New England, did some work on salinity tolerances, and others have as well. And I think basically the advantage they have is they're reptiles, and so their skin's pretty impermeable. They're not like frogs. You change the salinity and you'll knock frogs out pretty quickly. Well, with turtles, they're basically reptiles that have impermeable skin living in water. And so the ones that are affected by the salinity most dramatically are the ones that breathe through their bums. So the ones that have those cloacal gills and ventilate their cloaca, they're dramatically affected by salinity. So if you give a Rheodites a salinity bath to get rid of some fungal disease or something, you'll kill it. Whereas species like Longicollis, they just shut their little clackers and they don't open their mouths, and they swim around and feed and go on, and they're quite tolerant of the amount of salt that they do have to take in. And of course, then when they need to drink water, they can find access to fresh water, either floating on top of the salt water when it rains, or getting out and moving to a ephemeral pool that's not saline. So they have their ways of coping with salt that's both physiological and behavioural.
Okay, that turtles do seem to be fairly robust to it, like you said, with the skin impermeability. Seems like it's a sort of a huge thing, for helping them there. I sort of it brings to mind this other idea, the kind of effects of climate change on turtles, eastern longnecks, and Australian turtles in general. You've done a lot of work looking kind of how that on different aspects of the turtle biology that could be impacted by climate change. Curious what you think the worst impacts of climate change, and if turtles are adaptable enough to deal with the sort of prospective impacts of it.
Yeah, I think the biggest issue for Australian turtles is possibly not present in areas that are wetter. Is there's two aspects to it. Australia's sitting bang smack in the middle of a tectonic plate, and so there hasn't been much change to the river systems. They've been there for a long, long time, and so what happens is as the continent became more arid, you get restriction of some species down to single drainages where they've been sitting there for a long time, and I guess they're particularly vulnerable to climate change, rapid climate change, because they've got nowhere to go. So they can't. So they don't have a distribution that's spread across multiple drainages where they blink out in some, they reinvade others, and they've got a bunch of drainages where they can persist long enough to be able to accommodate the climate change. We've got a lot of species that are found in single drainages, and so if the climate changes dramatically, and the rivers dry up where they didn't, the water gets hotter where it didn't, then then basically they'll blink out. So climate change is an issue if we don't actively manage it in the context of some of these endangered species. So Pseudomajura imbrina, for example, like it used to be widespread in Australia in the Pleistocene. So they've got fossils of it up in Riversley, up in the Gulf country. It's now restricted by the progressive aridification of Australia down to the southwestern corner. Now, if it's got nowhere to go, if the climate changes dramatically and its winter wet, summer dry swamps disappear, it's got nowhere to go apart from becoming extinct. And so what do? We have to be very inventive about thinking about where this animal could live if only it could get there and help it get there. It's really crying out for some serious intervention in that sort of case, where you've got something that's been isolated to very small range, and now you've got climate change, rapid climate change coming in over the top of it. It's got nowhere to go but become extinct unless we intervene, look around the continent for areas where it could really thrive and assist it to get there. Now, that's very controversial, but in my view, that's something that's really necessary. And of course, the paleo data, the environments that the species used to occupy up at Riversley give you some insight as to what might be suitable for translocation of this species should the southwest corner of Western Australia become unsuitable.
Right. It's sort of case specific. Just to play sort of devil's advocate, maybe not even that much, but to kind of propose the opposing side to species translocations or creating new habitats sort of in the wake of climate change to house those animals, which is, I think, what you're sort of getting at. What is, I think a lot of people would say, well, they're not contributing overall. If we lose one species of turtle, it's not sort of a huge thing from a biodiversity perspective, an ecosystem functionality perspective. But what is sort of your argument for why resources should be allocated to protecting individual turtle species? And maybe just in general, why should people care about turtles?
Yeah, I guess that's an interesting question. A lot of people are, totally focused on the human species and its needs. And so when you talk about, what services does a turtle provide? Well, what services does a platypus provide? And yet we wouldn't want to see the platypus go extinct. But if it went extinct, I doubt that the river systems of Australia would suddenly, the ecosystems would collapse. But so we've got to, I think really it comes down to your philosophy as to whether we're caretakers of the planet and of the species that are here, and that we do sort of have a responsibility to ensure that these species don't go extinct. So that's sort of one philosophical point of view. The other one is that I guess people don't realise that all vertebrates are basically different experiments using the same genes and biochemical processes, and so you never know what's going to spring out at you. So one of the best drugs for treating diabetes type 2 came from studies of saliva in the Gila monster. Now, a few years ago, we would have said, oh, well, who cares if the Gila monster goes extinct? But you'd never know what's going to come out of studies of all these different little experiments with the same genes that we have that are driving our health and well-being. So even if you took a utilitarian point of view, you would still want to say, let's not let these species go extinct because they're a reservoir for opportunities to provide benefit for human health. Like we share 30% of our genes with a carrot, and we share like 99% of our genes with a chimpanzee, and probably more than 90% with a turtle. So, the dragon lizards I work on, we're working on the genes associated with sex determination in dragons. The mechanisms that determine sex in dragons that are influenced by temperature are the same pathways that cause human cancer, and the same inhibitors that they use to experiment on human cancer we're using to experiment on sex in dragons. That's how close we are in terms of the fundamental underpinning biology of our systems. And so every time you lose a species, you're losing a little bit of the opportunity we have to benefit ourselves in the future, even if you take that strict utilitarian sort of perspective. So you don't want them to go extinct.
It's an interesting point. I think it's a sort of a great perspective. As Anders Rodin, we had him on the podcast a while back, and he sort of said it was kind of a moral imperative to protect vertebrate life. And you kind of add this other perspective from your sort of the molecular and genetic background of it's also an information imperative to a certain extent. We're losing information about a system.
Yeah.
And that can be bad for us.
Yeah, exactly. So I hold the moral imperative view that Anders has, but I'm just trying to address the people who like I got rung up by a politician cold. I got a big grant to work on dragon lizards. I got rung up by a politician cold and said, why are we spending this money on dragon lizards? Why aren't we spending it on cystic fibrosis? Right. And so I had to answer that. And basically, it was difficult to answer a cold call like that, but basically I gave the sort of answer that I just outlined to you.
It's kind of a more concrete, like grounded in reality sort of like answer that sometimes you're going to need a simple reason for why is all of this justified?
Yeah, exactly. And it's got to be simple for a politician.
Yeah.
And it's got to be utilitarian. It's got to be like, what's it mean to my constituency? And so it's not enough just to argue the moral imperative, because there's a relatively small proportion of the population that think that way. And so you need to have answers for the broader elements of the community that are totally utilitarian. So.
Right. The information aspect of it is really fascinating. We had Brad Schaefer on recently as well, and talk sort of on your point with the Gila monster. He found something similar with the painted turtle genome, the SLC2A1 gene, and potential that has for diabetes therapeutics and such. And so that's just another example going towards your point in terms of sort of all biodiversity is important. That's kind of the utilitarian perspective, but it's necessary when arguing it.
Yeah, I mean, if you're going to get into that sort of stuff, I guess the aging, like aging is a big thing. The one things that billionaires can't do is extend their lives, right? We all die, and so there's great interest and a lot of money being directed towards aging research. And who's living long? Like turtles. And so if we can work out the mechanisms by which turtles have such incredibly long lives, that might give us some insight as to what we would need to manipulate to extend human lifespan. So that's a sort of utilitarian sort of perspective that can be important. I think so, and can deliver the goods. So there are a lot of species that don't have cancer at all, and turtles aren't among them. But there are a lot of species that don't have cancer at all. And by studying those species and studying how their genes interact, and the same genes as in us, how their genes interact, to work out how come they don't have cancer and we do, it's a big thing. So you'll see organisations like SciLife Labs in Sweden. It's a medical oriented organisation, but you'll see now it's biomedicine and biodiversity that they're studying. Why is the Wellcome Sanger Institute so interested in sequencing the genomes of all life in Great Britain? They're interested in this because of the payoffs that come from better understanding of the genomics of life, of which we're part. And so you don't want to be destroying your biodiversity willy-nilly without an appreciation of what you're actually losing in terms of opportunity.
Right. And so going back to something you mentioned early on, in terms of studying the sort of sex determining mechanisms in Pagona and turtles as well. We can sort of get into that. I think that's a really fascinating area of work. You've done quite a bit of research in, and I think a lot of people are familiar with the kind of high school biology conception of mammalian sex and meiosis, and it's very sort of the same thing conserved across mammals, and it's kind of a streamlined process that we learn about. But once you get outside of the mammalian classification, things become a lot more complex, and you get genetic sex determination, environmental determination, and all sorts of variations within that. Maybe you could give us a background, give people a background on how variable sex determination is outside of mammals, and why it's particularly interesting in turtles.
Yeah, so I guess the mammals are the odd man out in many ways. So first of all, they have a fairly conserved sex determining system. You know, we've got XXXY chromosomes. The males have the XY, so they determine the sex of the offspring, in the sense that the offspring that get the Y from the male are males, and the ones that don't are females. So it's a fairly relatively simple system that's portrayed in the textbooks. The mammals have their own variability. So there are some animals that don't have a Y chromosome; they've only got an X chromosome, and there are others that have multiple SRY genes on their sex chromosomes. So they've got multiple SRY genes, which some of which have been co-opted for other purposes. Like it's not completely simple in the mammals, and there's a lot of work yet to be done to understand that. But in the reptiles, there's a remarkable diversity. So we got parthenogens, species that produce only females. There's no males involved, and they perpetuate through parthenogenesis. That can be facultative parthenogenesis. So if you take a big sort of brannid and you put a female in isolation on an island, for example, then they'll throw offspring in the absence of a male. Some of those offspring will be male, so they can perpetuate themselves in that way. And others that's just been found are actually parthenogenic inside inside one clutch. So some of them are parthenogens, some of them are sexual. So you've got that parthenogenesis side to it. Then you've got TSD, which was discovered 50 years ago in the dragon lizard in Africa. You've got TSD, where temperature determines the sex, and there's no sex chromosomes at all. And that's really been shown to be widespread in turtles, not in Australian chelids, but widespread in many different groups of turtles. And that was met with disbelief when it was first came to the attention of science, but now it's well entrenched. But the fundamental understanding of how that works has eluded us for that 50 years, and it's only recently that we've started to get insights into how that works. And then you've got species that have sex chromosomes, just like us or like birds, and temperature comes in and diverts the pathway. And that's what our dragon lizard does. So it's got ZZZW chromosomes like birds. Temperature comes in over the top and reverses the sex of the ZZ animals to become females. So the males get reversed into female phenotype by temperature. So you've got really a spectrum of different possibilities, going from TSD at one end, where there's no underlying genetic predisposition, to GSD at the other end, where you've got sex chromosomes, and then you've got sort of intermediate forms where you've got the interaction between genes and environment to determine sex, which is, I think, we haven't looked properly. I think when we look, we'll find that that's much more common than the endpoints of pure TSD and pure GSD. So it's a very fertile field for research.
Right. It's easy enough to sort of take the terms and kind of label those systems as such, and kind of pass over that. But so much to think about when you actually think about the fact that some organisms don't have sex chromosomes, and the temperature is toggling with something at some point in incubation. Most turtles, that is sort of developing the sex organs. That's kind of a fascinating thing to think about, and it requires a lot of cutting edge technology, and you've dealt.
Yeah, well, it's frustrated science for a long time because, like you say, it's something that gets toggled by temperature, but it could have been anything. So it could be basically a gene that affects the passage of an effector molecule across the nuclear membrane, some really subtle thing that's affected by temperature that changes the sex. So you don't know where to look, and it could be anywhere. And what's happened recently, though, through the work of Blanche Capel and through our lab, is that it now appears that there's some really ancient mechanisms of gene expression machinery, really ancient mechanisms of gene expression machinery that are involved in giving temperature its effect. And so basically what happens is you've got these genes that govern whether the chromosome opens up for expression or shuts down. So not all genes in your cells are expressed at all times. So what happens is that some of them are expressed, the chromatin unfolds and allows the machinery to get in to express that gene. And the genes that affect that unfolding and folding are temperature sensitive. And so basically the temperature's affecting the expression of genes by making them available or unavailable for expression, which determines the sex. And so this is a mechanism that's really ancient that goes way back to yeast. And what we found is that there are some intron retention events in mutations in those genes that are very specific and are found in turtles, crocodiles, and dragon lizard. So they're very specific mutations in those genes that are ancient and have been maintained for 300 million years, right? And so they've got to have some really specific function. And so we've been looking very carefully at that function in terms of how it determines sex. And Blanche Capel's lab's been working on the same thing in the turtle. We're working in the dragon, which is a sex reversal scenario. She's working in the turtle, which is a TSD scenario. And we're both converging on the same sort of mechanisms that are involved in sex determination. So you can get your teeth into this because once you identify the mechanism, and if it's common, then you can get your teeth into it. Whereas before, it could have been almost anything that was doing it, and it was intractable. So it's a big advance in the last 3 or 4 years in terms of the direction research is taking to unravel how temperature influences sex in turtles. Exciting.
So it's sort of a restructuring of the genome almost, and temperature can intrinsically influence kind of the chromatin fibers that control a lot of that structure. And then that leads to differential expression, and that differential expression at a cellular level, or a sort of the nucleus at a nuclear level, how does that translate to the sort of development of each sex? What are the protein pathways and the transduction pathways leading to that? Is that known or?
Yeah, so the pathways that dictate sexual differentiation, that is the differentiation of the testes and ovary, are fairly conserved and fairly well known. So basically, if you look at a testes in a rat, or a testes in a human, or a testes in a turtle, or a testes in a lizard, they're very similar in structure. And so you'd expect that those gene pathways are fairly well defined and very canalized. So the pathways that actually result in the differentiation of the gonads are conserved and are fairly well known. But at the top of that sort of cascade, a decision is made: am I going to be a boy or am I going to be a girl? And it's that decision, the sex determination mechanism, that's really poorly known. And so in mammals, the SRY gene on the Y triggers a cascade that leads to a testes. In the absence of SRY, the cascade produces an ovary. So we know it's SRY in the mammals. No sex-determining gene has yet been discovered in a turtle, or indeed in any reptile that's been actually demonstrated. There's some good candidates, but none's been demonstrated in reptiles. And of course, it's complicated by this this temperature influence, and how does that influence that upper decision-making process is the interesting story, I think.
And this sort of falls in the realm of epigenetic control. Is that the right way to think about?
Yeah, temperature-dependent sex determination is epigenetic by definition, right? Because what you're saying is that there's an influence that's outside the genome. So it's epigenetic by definition. So we're talking about epigenetic processes that lead to the expression or non-expression of a key sex gene like DMRT1. That's what you're talking about. So epigenetic processes by definition, yeah.
From an evolutionary perspective, why would temperature dependent sex determination be something beneficial? Why would that evolve?
Yeah, well, I mean that's a really interesting question. And I guess turtles, create a confounding because they're so long lived that the sex determination in the nest of a single nest in a species that's going to be breeding for 60 years and laying 3 or 4 clutches a year in some cases, like how does that matter so much, I guess. So but there are short-lived species like dragon lizards and perhaps some turtles where I guess there's an advantage to being male under some circumstances. There's an advantage to being female under other circumstances in the nest, and they can't be predicted by the mother at the time she lays the nest. And so the decision on what sex they're going to be is deferred until such time as the information is known as to whether it's going to favour males or females. And then that decision is made by temperature, which is directly correlated to the fitness benefit that is afforded to either male or female. So this is the Bull-Charnock model. So basically, they defer the decision about making boys or girls until the information is available within the nest as to whether the advantage is going to be accrued by males or by females, and then the decision is made at that point. So it's made by temperature. So for example, if you're laying multiple clutches in the year, the later clutches are going to hit winter before the earlier clutches. So the ones that come out earlier are going to have a growth spurt before they enter winter. The ones that come out later are not. And if there's an advantage of being large if you're a female, and not so much an advantage of being large if you're a male, then obviously if you lay your nest early in the season, it's better to produce females because it's going to get that initial growth spurt. And the ones that are laid later, it's better to be males because they don't need that growth spurt. So you can see that there could be a selective advantage to it. I think it's really difficult to get in your teeth into these scenarios in an experimental way. And one of the results of our work has shown that you can get sex reversal by climate change without any selective advantage at all. It's just basically frequency-dependent selection that drives the transition. You don't need to invoke any selective advantage one way or the other to evolve from GSD to TSD. It just happens because of the overproduction of one sex through sex reversal, leading to driving down of the sex chromosome through frequency-dependent selection, and ultimately the loss of the sex chromosome, and you move to TSD. And that all happens simply because of that frequency-dependent selection. You don't need to invoke selective advantage to drive that process under conventional selection. And but once, of course, it moves to TSD, then conventional selection locks in to maintain it. So the question becomes, what selective processes maintain TSD once it occurs, rather than what are the selective processes that drive the transition. So that appeared in an article in Vows in Nature in 2015, if you want to look that up.
Yeah, it just in terms of a question for overall biodiversity, it's fascinating. and to think about why it seems to make sense why there'd be an advantage to offset the determining factor. And maybe even, I'm curious what your thoughts about this are, but it doesn't seem like in every case an even sex ratio in populations is beneficial. There may be times when resources demand more females and males, or vice versa. And maybe that could be something that temperature is sort of modulating.
Yeah. Yeah, it could be. I mean, selection operates on the individual. So you've got to think, what's the advantage to the individual? So it might be an advantage to a population to have more females because the effective population size is larger as a result, because you don't need even sex ratio to perpetuate if the males are mating with multiple females. So, why do you need a 50-50 sex ratio? But you've got to look at the individual and what selective advantage is there. And that tends to lead to a 1:1 sex ratio over time, because the animals that produce more of the rarest sex have an advantage over, in terms of number of grandchildren, over the ones that produce more of the common sex. And so you get this selective advantage that quickly drives the system towards 1:1. But there's a caveat on that. When the environmental conditions that determine the sex are also influencing offspring survivorship, then the 1:1 is no longer the sex ratio that's optimal. So the sex ratio can be quite different from 1:1 when the environmental factor that's influencing sex also influences offspring fitness in other ways. It shifts from 1:1 to something else. And so expecting 1:1 in reptiles with TSD is not really a sensible null hypothesis if temperature is also affecting offspring fitness in other ways. So you have to qualify it. There's not much in the literature on that. I think James Bull pointed that out in some of his early papers on sex determination, but no one's really picked up on that since.
Yeah, it's something you pointed out too that's interesting about it is with the sex reversal and sort of the evolution of genetic sex determination and switching between environmental sex determination. That there's almost sort of inherent, the idea there is that one can come from the other, and you've seen this occur fairly rapidly. And so if you just have that frequency dependent selection, that transition could be something in the system that's kind of inherent, and that sort of rapid turnover, maybe it's just an adaptable response to different conditions.
Yeah, so in mammals, because they've had their sex determining system for so long, you can't do without your Y, right? You can't produce spermatozoa without a Y. It's basically locked in because of millions of years of refinement. And in reptiles, that's not the case. And one should ask, well, why isn't that the case? And one of the reasons why is that I think temperature-dependent sex determination, TSD, is a disruptor in an evolutionary context. So you've had this turnover, and it's self-reinforcing because there hasn't been time for any lineage to evolve to lock down the necessity of having a Y chromosome. And you've got a lot of species that have homomorphic sex chromosomes, so that sex chromosomes haven't differentiated and they haven't locked down because they haven't had time to lock down, because you've got this evolutionary churn that's been going on with TSD coming into the equation. I think that's a really interesting sort of thing that could be developed more in the literature, that idea of TSD being a disruptor and leading to maintenance of the diversity of sex determining mechanisms in reptiles. Now, you might have snakes, for example, that, if you separate the pythons out, snakes tend to have ZW chromosomes like birds, highly conserved. Maybe they have locked down. So maybe some lineages of reptiles have locked down into a particular type of genotypic sex determination, but reptiles as a whole haven't.
And so with regard to sort of the temperature dependent sex determination, you've looked at sort of how that works, the dynamics of that in loggerheads and other turtles. And something that I found fascinating with that is a lot of the early studies that looked at sort of classifying if it was TSD or GSD in a species were more sort of empirical and lab based, and you've sort of looked at this in the wild, and something that's inherent in natural systems is some level of temperature variation and variability. It's not always going to be a constant temperature exposure, and you've seen that it's more a function of the amount of development that is occurring at a certain temperature than it is that constant exposure. Maybe you can speak to how variability in temperature influences sex ratios of turtles that are TSD.
Yeah, so I guess like most of the early experiments were done at constant temperatures. And so you do the constant temperatures and you show that you get 100% males at one end, you get 100% females at the other end of the spectrum, and you get mixed sexes in a very narrow range. And so it's natural just to translate that into trying to interpret what happens in nature. But in the natural nest, the temperatures are all over the place, so they go up and down each day. So in pig-nosed turtles, they'll drop down as low as 18 degrees, and they'll go up as high as 45 degrees. And so they're going up and down, and you get rainfall events. You get all sorts of impacts on the temperatures in those nests. It's quite complicated. And I think one of the advances that I made early in the piece was to show that it wasn't just the mean temperature that was influencing sex, but also the variability. And in fact, I was able to show that you could switch from 100% males to 100% females, not by changing the mean temperature. You could keep that constant, say at 26 degrees, but you could flip the sex from 100% male to 100% female just by changing the amount of fluctuations around that mean. And so it's both the mean and the variance in temperature that determines sex. And then I guess I should add that the way I got into this, and it's sort of interesting. I decided to become a scientist at 14 when I read some of Isaac Asimov's factual books, and he talked about the milestones in scientific life, which is normally a drudgery most of the time. And one of them is it's that funny moment. You know, you go, you're looking at something, you go, oh, that's funny, and then because it doesn't fit with your understanding. And so Graham Webb did the experiments on the pig-nosed turtle, showing that they had temperature-dependent sex determination. He characterized with constant temperatures, exactly how it worked. I went out into the field, started measuring the temperatures in the nest, and found that the temperature that was the pivot point was 1.5 degrees different from the temperature he found in the lab. I went, that's funny. And of course, I trusted his work impeccably. He's an outstanding scientist in terms of being meticulous. So I trusted his lab work. I trusted my own work. So 1.5 degrees. Why was that? And it turns out it's because of the fluctuations in the nest in addition to the mean. And then I worked out using calculus and other things how to characterize that mathematically. And then I had really good, precise expectations in my experiments. And I could do my experiments on a loggerhead turtle to show that those expectations were indeed met. And I could show quite clearly that the fluctuations dictated sex determination, and I could say why, because I understood the mathematics, so I could say why it was. And it's because an animal spends more of its life above its mean temperature than below its mean temperature if you're a reptile. So when you're hot, your growth and metabolism is going like the clappers, and when you're cold, you slow right down. And so you spend more of your life above the mean temperature than below it. And so above the mean temperature has much greater influence on your sex ratio as an embryo than below it. And the mathematics sort of encapsulates that and makes really precise predictions that enable you to take the lab work and translate that into field work. So that was sort of early in the piece of my career that I sort of did that work. I'm still excited about it because it was a that funny moment, which doesn't happen very often. And I could have dismissed that 1.5 degrees as just, errors. But it was a that funny moment. And it persisted, until I thought, well, that's funny. And then the next moment is, oh, I know why. So there are a couple of really bright moments in a scientist's life. It's a that's funny moment. and then there's a realization of why, and all the rest is drudgery, like just hard work. But you're sustained by those little moments in your life.
In one of my past calculus classes, we had to brainstorm areas where you'd use different methods of integration, and if I had known about, well, I did know about it, but I wasn't thinking about it. But it's admirable that you use trigonometric integration to solve that question. I just thought it's cool to see, because a lot of times people sort of pass that whole sort of area of science and mathematics off as kind of something that's not useful much, but you used it to answer a question related to sort of how development is related to fluctuation, right? And it's cool. It was cool to see.
Yeah. So my undergraduate degree's in pure maths, right? So like I wandered off. I saw turtles as this esoteric little hobby, and I did the maths. And then I went up to Managrita with Harry Messel and Graham Webb working on crocodiles, and I realised there's another side to life, and that you could actually study your passion. And so I finished the maths degree, and then I did extra undergraduate work in zoology, and then did my PhD in zoology. But I still had the maths and the understanding of calculus, and so it was an obvious thing to do. And nowadays, of course, you don't need calculus. You've got the computational power just to iteratively sum across the temperature traces to get your answers. But back then, basically, analytical approaches were the way to go.
Right. Would there be? So you mentioned one of the limitations of what you did is that you couldn't incorporate sort of random fluctuations and years where you had a lot of rain and random sort of the ends of the spectrum of stochastic events into the model. Do you think that's something now you could do with more complex algorithms?
It's challenging. So what the model does is it says that if you take a given day and you look at the temperature cycle, you can say what if that day was perpetuated across a whole thermosensitive period, what sex you'd get. That when you have variability through the thermosensitive period, so you've got a certain regime at the beginning, then you've got a rainfall event, then the temperature's warming through the season, all of that. The model doesn't really address that. The way to address that is to look at the contribution of each day to a particular sex across the thermosensitive period, and then match that with the sexual outcomes, and then try and come up with some sort of correlative relationship as to what your expectation would be based on a trace. It's not as satisfying as the mathematics that apply to a single day, but it can be done. But you can't apply the model, as some people have done, across the whole thermosensitive period and try and predict the sex. You've got to apply it each day, do a trend on the likely sex influence across the whole thermosensitive period, and then make your judgment. Of course, if across the whole thermosensitive period it's all up in the female domain, you're going to get females. It's all in the male domain, you're going to get males. But if it crosses over halfway through the thermosensitive period, well, who knows? That's the problem, and that hasn't been solved. Yeah.
That's sort of an open question with more complicated algorithms, and
someone's going to solve it. I mean, it just needs a work where you've got a lot of nests, you've got a lot of temperature traces. Make sure you've got the probes at the top, bottom, and middle of the nests, and you've got a lot of nests. You know what the sexual outcome was, and you can work through that, but it hasn't been done satisfactorily yet. I mean, I've tried to do it with a pig-nosed turtle, and I have a paper that is ready for submission that does that. But I haven't submitted it yet.
Is there some way? I've done some work with Bayesian modeling and such, and just curious what you think, if there'd be a way to incorporate that into that sort of iterative trigonometric modeling.
Yeah, I think it lends itself to that. So Bayesian modeling. So you've got the the standard probability that people use in t-tests and that. And then you've got maximum likelihood. And then you've got the Bayesian modeling where you can feed in priors. So based on, your previous data is collected to that point, you can set up some priors and feed that into your model to get better predictions. And so it certainly lends itself to that in the same way as the original application of firing a cannon at a target and in a war and finding you miss by a certain amount, and then you feed that back into the Bayesian modeling, adjust it, fire again, miss by a certain amount, feed it back in until you get better and better at hitting your target. So that sort of analysis is most appropriate for that question. Yeah, definitely.
Yeah, some way to incorporate some level of the stochasticity that in temperature over a season. Maybe there's some way to attach a statistical level to the sort of linear aspect and the trigonometric aspect to it.
Yeah. It needs a young mind. Needs a young mind. I turned 70 this year, so it definitely needs a young mind to look at it, because your math security disappears over with age. So.
Yeah. Well, it's something that the listeners and anyone that's interested in that sort of thing, it's an open question in terms of looking at how seasonal variability influences the models that exist for determining kind of how the variation in temperature influences sex. That's something there. So another thing too that you mentioned, the pig-nosed turtles, that's a really fascinating one, and you've got field experience with those. Curious, just what are some of the experiences you've had with them? Maybe I know you've worked in the Kakori region of New Guinea, and I'm sure you've had some adventures there.
Yeah. So so basically, the pig-nosed turtles are iconic species for obviously for Western communities, in the sense it's the last remaining member of its family, and so its conservation is the priority is high. In the Kakori, it's an iconic species because it's treated like a fishery, right? So the meat and the eggs are highly valued by the local community, and they're an important component of their subsistence environment. So both from a conservation point of view, we don't want to see it go extinct, and from a fisheries point of view, you don't want to see it go extinct. So we were working with the support of Oil Search and ExxonMobil. We were working with the local community to assist them in terms of sustainability of the environment overall, using the iconic pig-nosed turtle as a sort of an umbrella species. A lot of interest in it in the community. Marshal that interest to try and engender some urgency about ensuring that their environment is sustainable into the future, and also just conveying that concept of sustainability to people who are living hand to mouth with a 4-day focus on whether they're going to be able to feed their kids. So it was a really interesting project. It involved a lot of community education, community capacity building. It involved a lot of learning about what the community's perspectives were and what they wanted. And then being able to bring science to the table so that they can make their decisions about achieving what they wanted with the best available information at hand. So it's a really interesting project, and it hasn't finished. It's still going. There's a team of people up there still working on this with the support of a broader base of funding agencies now. So it's great. It was great stuff.
Do you think that species can be sustainably harvested for subsistence purposes? Is that something?
I mean, we were fortunate that Mark Rose was doing a PhD project up there in the 80s, and that he gathered information on the number of eggs that passed through the markets, the number of turtles that went through the markets. And he took 3 villages and he monitored the number of eggs consumed a number of turtles consumed in those 3 villages over a couple of years. So we had a good baseline data, which he kindly provided us. And then we went and repeated that 20 years later. And we showed that the population had declined by about 52%. So up until that point, the community didn't accept that. They thought that the turtles had been put there, by the grace of their ancestors to use, and that they could use it in perpetuity. The old people were saying there used to be a lot more turtles around. It's like me saying to my grandchildren, times were better when I was your age. They just dismissed it. By engaging the community in these surveys, they could actually see for themselves that the turtles had declined. It gave credibility to the old people that were saying there used to be a lot more eggs, there used to be a lot more turtles. And so the community then became aware that they had a problem. And that was a big achievement, I think, just convincing the community that there was actually a problem and that they needed to address it. The sustainability message was a challenge. I don't know, I probably lost the thread of the question here, so maybe I'll just stop there on this question.
Yeah, it sort of makes sense. Or it's a question of, I think a lot of people, obviously that's an area that isn't, there's not a lot of income there. So harvesting turtles is important to just keep the population healthy. And if you can't provide an alternative livelihood, people morally want to change, but there's sort of no incentive to do that.
Well, it's cultural. So if you provide a lot of resources, so if you provide a lot of funding for petrol and boats and things like that, then they'll more effectively harvest those turtles. So one person can flip, 20 turtles over on a nesting beach. One person can harvest, 20 animals, pile them into a boat, take them back to a village. So they're very effective at harvesting these things. So the fact that they're becoming more affluent in a financial sense in terms of money means that they can afford boats, they can afford the fuel, and the cultural imperative to harvest these animals is still very high. So I don't think that it's ever going to go away that they harvest these turtles. So I think one of the big areas where we have some hope is that different religions have different views over the turtles. So the Baptist religion in PNG, people who follow that religion won't eat the turtles or their eggs. And so if you go to the Turama River, the turtles are there, present in historical abundances. And the nesting turtles, it's phenomenal. The nesting of the turtles in the Turama River is phenomenal because the community doesn't harvest them. But in the Kakori, they do harvest them. And so, they've dropped down like any fishery would. It drops from the sort of pristine, unharvested levels down. Perhaps it could go down as far as 30% before a conventional fishery would become concerned, right? They expect the standing crop to drop when you institute a fishery. So it's dropped down in the Kakori. But the good news is the turtles are moving right across the Gulf. And so you've got these patches where they're not being harvested that can feed through into the areas where they are being harvested. So there's some hope that the patchy nature of this exploitation in PNG is leading to the sustainability in the long term of the species in that region. So that's a hope. If ever it got where they were exporting eggs and meat, as they do from sort of Papuan, Indonesian Papua, then it brings in a whole new raft of considerations that need to be made. But at this stage, it's not the case in Papua New Guinea.
Right. Okay. And yeah, it's interesting to think about that conservation perspective. Maybe we can sort of switch focus a bit to something else that you've done for many years, sort of looking at the phylogenetics of Australian chelids and turtles in general. This has kind of been a fascinating thing, and you've got an interesting perspective that Dr. Schaefer also had with respect to sort of global turtle phylogeny. You've analyzed the question related to Australian chelids, how they're related in many different capacities over time. And so you've seen those relationships change with different analytical methods and such. But in your initial attempt to put together the Australian turtle phylogenetic tree, what did you find? What were the things that were surprising to you? And how has your understanding of that tree changed with time?
Yeah, so I think the most interesting thing was that people had in the past, based on morphology, had grouped the long-necked turtles of South America with the long-necked turtles of Australia into a single clade. And I think with the molecular work, it was quite clear that that wasn't the case, that the Australian radiation is a single clade, and that the South American long-necked turtles are independently derived their long necks in comparison with the Australian turtles. So the long-necked Hydromedusa in South America and the long-necked Chelidina longicollis in Australia aren't in the same clade. They've independently evolved their long necks, or they're plesiomorphic. The long necks were ancestral, and they've independently retained them. So that was, I think, a really interesting result, and it was borne out by Pritchard's studies of how the shell forms to accommodate the longer neck, and the shell in Hydromedusa has accommodated, and anteriorly has accommodated a long neck in quite a different way from the Australian Chelidina. Right. And so they are, the morphology supports their independent evolution as well, if you look at Pritchard's data and weigh it against the other morphological data that was provided. So that was one result. I think the other result was that there were all these really deep ancestral lineages in Australia. So Australia presumably had a really diverse fauna, and then with the progressive aridification of Australia, then that's been maybe not lost, but probably there's elements of it lost, but it's certainly been contracted down with species now occupying single drainages. And so finding out that the pet shop turtle, for example, which is only known from pet shops, wasn't a pet shop hybrid, but actually a really distinct and ancestral lineage of Australian turtles. And this was done before it was discovered to have come from the Mary River by John Cann through his incredible detective work. Found that it was in the Mary River, but we'd already done the genetics and showed that it wasn't a pet shop hybrid. It was a really distinct and ancient lineage. And the other surprise, I think, that came out of it was that Legler had Myachilles georgesi and Myachilles purvisi as the same species. And sensibly so, because they're very similar. There are coloration differences. The sort of Manning River turtles are brighter color than the most beautiful turtle in the world, the Myachilles georgesi. What to say? But anyway, color's not everything. So basically, these were seen as the same species, but the genetics showed that in fact they were ancient, and that Myachilles belli and Myachilles latisternum have emerged from within that clade between Myachilles purvisi and Myachilles georgesi, right? So they're ancient, and the fact that they're so similar gives you a really rare insight into what they looked like, what the ancestor looked like. So they haven't changed over millions of years. They've given rise to Bell's turtle and to the common snapper, which has changed quite dramatically from them, but they haven't changed at all in that time. And so they represent the morphotype of the ancestor, which I think is really interesting, because you get that glimpse into the past of what this thing looked like way back then, and then as the ancestor to latisternum and belli. So it's quite an interesting thing that came out of it. I guess then I've moved more to species delimitation and away from the phylogeny, though it's obvious that there's going to be some massive advances in phylogeny with the new genomics revolution that's going on. So we'll have genomes for all these turtles in not too long and be able to have a really good look from a whole genome point of view at their evolution. And the area that's opened up by that is the fact that we'll be able to look at structural variation in the genome, not just sequence variation. And structural variants, the shared inheritance of a structural variant that's really unusual, is much more powerful than just sequence variation. So I reckon there's going to be a revolution in our understanding of the phylogenetic history of turtles globally, which will include the Australian ones, which I think will be really interesting. So I've moved more to sort of species delimitation and looking at the much more finer boundaries between species of turtle. And the paper on the southern Emodura that came out in 2018 is the first of a series of papers that I'm hoping to produce using SNP markers, which are the same markers that Ancestry.com uses and 23andMe. So basically, we're DNA fingerprinting the turtles and looking at their fine-scale relationships across the landscape. So we're coming at it from a different direction. We're coming at it from the population genetics direction rather than from the phylogenetics direction. And the two meet at some point. But because it's much more fine scale, you've still got the process of speciation in progress. And so it's a much more fuzzier picture, a lot harder to get your teeth into.
Right. That brings up something that's really interesting. And I'm curious about kind of how you articulate this. Someone asked me this question recently. It's, and in my mind it's there, but it's sort of hard for me to put into words in terms of what the difference between phylogenetics and population genetics is with respect to species delimitation in terms of the types of analyses you're using and that sort of thing.
Yeah, there's different camps. So some people have basically said that lineage is a species, and so if you can demonstrate that an entity, say a population of turtles that's in a particular region, is on an independent evolutionary trajectory, that is, it's a lineage, then that's a species. And so there are a lot of people that take that view, so they use phylogenetics to delimit species because the phylogenetics identifies these lineages and then the lineages are species. And that's quite a common view, I think. It's not one I hold, but it is quite a common view. The second view is that you can have lineages within species. And so species aren't just defined by phylogeny, they need to be defined under the biological species concept. So they're entities that are on independent evolutionary trajectories that don't just arise through geographic happenstance. They derive because they're reproductively isolated. And so if they subsequently come together, they'll still persist. Whereas if lineages come together, they can obliterate each other, right? Because they're not necessarily reproductively isolated. So I've got a more traditional view. Now, the biological species concept's got warts on it because in sympatry, no problem. You find that there's two genetic pools in the one lake. Even though they're morphologically identical, you say they're different species. No one argues. But in allopatry, like you expect differences to accrue between lineages within a species because of the geographic isolation. That doesn't mean that you call them different species or subspecies. They're just different lineages within a species, which is what you'd expect. And so where the population genetics comes in is you ask yourself the question, you set up a null hypothesis. You say the null hypothesis is that these are all one species. And then you look at the evidence and say, does the evidence is the evidence sufficient to reject that null hypothesis? Have they got the opportunity for gene flow? If they have, and they're still distinct, then that rejects your null hypothesis that they're the one species. And so, you can use the population genetics to examine whether or not there's gene flow across a landscape, whether it follows the pattern that you expect from their opportunity for exchange of individuals, or are there abrupt breaks that are not consistent with their ability to exchange across the landscape. So it's coming at it from a different direction, and then you decide your species based on whether or not they've accrued fixed allelic differences in a context of potential gene flow. Now, if they're completely allopatric, you've still got that old problem of, are they species or are they just lineages within species? So it doesn't solve it if they're completely allopatric. But if you study something like the southern immature across a landscape, you sample thoroughly across that landscape, there's no breaks, and then you look at the distribution of fixed differences across that landscape in the context of their opportunity to exchange, and you find there's a big set of fixed differences between two entities in there, you've got two species. If there's no fixed differences across that landscape that aren't representative necessarily of the ends of a cline, then you say you've got one species. Strictly, you say you have no evidence to reject the null hypothesis that there's only one species there. So that's the approach I take, and it requires a lot of sampling. So you can't have gaps, because if you have gaps, you're going to have an artificial break in the sort of architecture of the genes, alleles across the landscape. So you've got to thoroughly sample, and then you address that null hypothesis. They're all the one species. And then if you've got the evidence to reject that, then you've got a couple of species. If you don't have the evidence to reject that, then there might be species there, but you just don't have the evidence to show them. And so you don't change the status quo. So it's a sort of a very sort of Popperian approach to it, using PopGen, coming from a different direction that the phylogenetics come from. So once you've got your entities that are genetically distinct with fixed differences, so they're diagnosable, then you apply phylogeny to work out how they're related to each other. You see what I mean? So you don't apply the phylogeny to individuals and then try and work out where you're dealing with diagnosable entities and then define your species. You do it the other way. You use the fixed difference analysis to define your species. And once you've worked out what the diagnosable lineages are, which are species and lineages within species, then you can apply your phylogeny to see how they're related in a bifurcating tree. So I'm on the outer on this. Not everyone agrees with this approach.
Right, and it's Australia in particular has had a pretty turbulent taxonomic history when it comes to turtles. I'm curious, and a lot of that's based on kind of morphological work that's been done, and it just seems to be, compared to other places, it's been really turbulent. So I'm curious what your take on that is, and why that's been the case.
I think it comes from a frustration of lack of progress on the morphological side. So we had John Legler, who was working pretty intensively on the morphological side, and he and John Can published the Rheodites description, the Elusor description. Excellent work, published through appropriate channels. And then, of course, I guess bogged down by the exactitude of his science, he died before he published anything else. And so that's really unfortunate. And there's a lot of frustration in the herpetological community about that. And instead of really sort of people are rising up through the ranks to replace Legler, like Scott Thompson has, and there are others who have raised up through the ranks. Instead of doing that and addressing the issue from a scientific perspective, we've had a breakout of people who are throwing their ideas out there outside the mainstream scientific channels and the cross checks that are present in scientific publication, and that's causing a lot of grief. So basically, without mentioning any names, there are people who are publishing in their own journals. Like I say, their own journals in the sense that they're the editor, and they're also publishing. Although they claim that it's peer review, it's really hard to see that it's peer review that meets the norms of scientific publication, which is independent, rigorous, expert rebuttal of your ideas and testing of your ideas before publication occurs. That's a really important hallmark of science. There are other cases, so we're talking about hundreds, if not thousands, of species being described here, right? Like hundreds of species being described, but without a strong scientific foundation. And so the difficulty for a scientist is that if someone puts a concept forward like that and it hasn't gone through peer review, so it hasn't been that pre-publication scrutiny, then there's a massive amount of work if you want to follow through the scientific process to rebut it. So it's easy to throw an idea out there on a napkin, really hard to rebut it if there's no foundation for what, no scientific foundation for what was put forward. And this is the conundrum, I think, that there's a lot of this stuff being published without a strong scientific foundation, and yet the scientists are expected to drop tools and start focusing on generating evidence to show that those propositions are without foundation, and people aren't willing to do that, and so my view is, and we've really addressed this through the Australian Society of Herpetologists, is that if you don't publish through an accepted scientific channel, then people can elect to ignore your work. And so that's very contentious, because the ICZN, the governing body over nomenclature, will argue that these that when someone puts a name forward, they have precedence. And then, of course, what that means is that the biology that they've attached to the taxon that they put the name to has to be considered. And when that's done through some sort of personal publication without any cross-checks, that creates major problems that don't plague any other discipline of science. So when someone proposed, as they did in Queensland, their Premier picked it up, that there was a new method of driving cars based on, that was solely driven by water. So you put water in your tank and it drives your car. It's revolutionary, except it wasn't published anywhere. And so, it was ignored, right? When someone puts forward a proposition that they've discovered cold fusion and they publish it, it goes through peer review, so there's pre-publication scrutiny of the evidence, and then it's published, the whole scientific community swings behind that, even though it turned out to be wrong. The whole scientific community swings behind it. They spend millions of dollars to try and confirm or refute that proposition. That's the way science works. If those people had published their cold fusion idea on a napkin or on a milk carton, even though it meets the criteria of the ICZN, say, it's just resoundingly ignored by physicists. So the publication process is really important to me. And if you want to put forward a new species, do the hard yards, gather the evidence, put it before your peers to scrutinise it and rebut it. The onus is on you to prove that the species exists at that point. Once it's published, the onus is on everyone else to disprove it. So the onus switches at that point. And that's a really important part of the process, the peer review process. And these guys are bypassing that. So they're putting it out there, and then it's up to the scientific community to rebut it. But it hasn't been through that process of putting the evidence before experts in the field. So it's a problem for us in Australia. I think it arises because of our inherent and deeply rooted distrust of authority. So So Australians are particularly known for that, and so we tend to have these people come out of the woodwork who just don't like authority, and they see the scientific community as being this elite authority, and who are they to be dictating these questions? So there's coming out of that sort of mentality, I think, and I can understand that because I've got a deep disrespect for authority. So anyway, so it's a problem for Australia. We've addressed it in Australia by having a positive list of journals, which is the Web of Science journals, plus journals produced by museums, plus some others that we've added as a society. If it's not published in there, we can all choose to ignore it or not. Whereas under the previous regime, we couldn't choose to ignore it. And now we're saying, well, we can choose to ignore
it. It must be frustrating, too, coming from your perspective, having looked at questions, and that's sort of just proposing names almost, a lot of that taxonomic vandalism. But from your perspective, you've worked for years on a lot of these questions using really sort of deep analytical, complex methodologies, and you've traveled all around Australia. I mean, thinking about the phylogenetic work that you did, you had sites you were sampling all over. And for someone to come in and just completely try to mess all of that up or add things that aren't at all with any sort of basis, it's got to be really kind of a frustrating thing.
Yeah, it's frustrating because scientists expect to be rebutted, right? So you expect there's a couple of processes. So when you submit something for publication, it goes out for independent review. The reviewers come back and they go over it. So that vertebrate zoology article that Scott Thompson and I put out recently on the Laverack aurum that went to the journal Vertebrate Zoology, it went out to rigorous review. It went backwards and forwards between the peer reviewer and us to get, and it's a much better manuscript as a result. And basically we had to convince those experts that our evidence was sound. Then then it gets published, right? So that process is time consuming and essential, and to have people sort of bypass that is the frustration. It's not that the work's being rebutted after all that work's been done. Natural thing that happens in scientific literature, like everyone expects that when you publish an article, someone's going to publish new evidence that suggests that maybe your ideas weren't quite right, and you expect that, and that's good and healthy, and you've been noticed. But when it's done outside the scientific literature, and we're forced by the ICZN to acknowledge that, it becomes really problematic, because a simple little thing. So I could describe a species in my own published journal that's characterized by a red stripe on the side of the face that's only present in the absence of light, right? Now, that character is acceptable by the ICZN. It's completely unacceptable by science because it's irrefutable. If it's only present in the absence of light, you can't detect it. So having a character like that's acceptable to the ICZN, can be published in your own self-published magazine, is irrefutable by science. We could spend an infinite number of dollars trying to refute that, and you couldn't do it. This is really frustrating that there's no scrutiny of the evidence before it gets published. And I've got a blog on this that talks about the onus of proof, and it talks about Bertrand Russell's teapot, which sits halfway between the sun and the earth. And someone proposes it's there. The onus is on them to prove it's there and provide the evidence. The onus isn't on others to disprove that it's there. So this is the problem. If they come up with a new species, the onus is on you to prove that it exists. And the peer review process is where you do that. Once it gets through the peer review process and it's published, then the scientific community assumes it exists. And then they gather evidence to sort of either confirm or refute that. That's the process. These guys are bypassing it. They're just throwing species out there without that pre-publication scrutiny of the evidence. And so they're proposing these Bertrand Russell teapots all over the place, and we're expected to acknowledge that they exist, and then to prove that they don't. Well, that's not the way science works, and that's the frustration, I think. It's not that we're being criticised.
Right. It's just sort of vandalism. I think it's good to get that out there and for people to think about that but to me, it's interesting. I've always thought this should be the case everywhere, but the fact that there's sort of underpinnings societally in Australia where you get that sort of distrust of authority and such, it sort of maybe manifests in taxonomy a bit more than it does in other places. So that's interesting to hear.
Yeah, we're different. We're Americans are independent, and we're independent, but there's a difference in our independence. It's subtly different, the way in which we're suspicious of authority. I see in America there's a lot of distrust of authority, and that is a very subtle difference between the way that manifests in Australia compared to the way it manifests in the United States. And the way you're seeing it manifest in this instance, I think, in the way people are, happy to challenge authority in the way that these. I won't call them vandals because I just think they've got a different ethic. They're not scientists. Why should I expect them to have a scientific ethic? They've got a different ethic. It might be he who dies with most turtles wins. Who knows what ethic they've got? So I tend to avoid the word vandalism, but just they're not. They're not practicing science, so why should scientists take notice of it?
Right. Going back to some of the stuff that is interesting about the Australian phylogenetic work you did with the turtles, a lot of that is the stuff that gets put in the paper is obviously very interesting, but there's some level of that in terms of going out and sampling in the field that isn't pertinent to discuss in a manuscript. But I'm curious what that experience was like and going and getting the samples, the adventures you had, if there was anything that stands out as just something that was really interesting from doing the field work necessary for that.
Oh, I just love field work. I mean, I love being out in the bush. I sleep on the ground in a swag. It's just a fantastic experience being out in the field, and you've got a purpose when you're out there. So, I'm not the sort of guy who could go and lie on a beach for a couple of weeks and have a holiday. It's just if I don't have a purpose, and it's got to be a fairly strong one at any point in time, I'm not happy. And so you're in the field, you're enjoying yourself. I'm not exactly. I wouldn't say I'm a recreational ecologist, but I do really enjoy being out in the field and doing something that's got a real purpose to it. And I have had some exciting times. Brad Schaefer might have mentioned the exciting time and consequences of our trip in New Guinea, or he might not have. But I've had exciting times. So my wife is really tolerant. I've had situations where I've been measuring temperatures of pig-nosed turtle nests before data loggers were affordable. Where I had to go up the river every hour and 15 minutes to measure the temperatures. And I was up the river, and the brucellosis program was on, and the landowners and Aboriginal rangers drove 300 head of steer through our camp where my wife was with her sort of 4-year-old son and baby, and like they trashed the camp, and she was hiding behind a tree. The Aboriginal rangers the horsemen were trying to keep the steer away. They had these big horns. They were goring the horses in the chest, and like it was a complete mess. And I came back and putt back down the river and come back and find the camp was completely trashed.
Wow.
And there was my wife and 2 kids in the midst of it. That sort of, there's lots of things go wrong when you're out in the field, but we survive them, and later, it becomes a great story at parties, but at the time it can be quite challenging. So there's been a bit of that. Catching snakes has always been a passion. It's always an adrenaline hit. That sort of thing, like the field work, like reptiles. I'm a herpo underneath, but I also do research, and I've married the two, which is really pleasing to be able to do.
What was it like to work with Hemidura crepti on Fraser Island for your thesis?
It was fantastic. So Fraser Island's about 100 kilometres long. It's the biggest sand island in the world, I think, and it's got 40 dune lakes. And these dune lakes are all isolated, so they've never been connected to the sea, and so they've got really unique attributes, and they're acid lakes. And they're oligotrophic lakes, and so you've got these turtles in there. There's actually 3 species, but the Hemidura are really interesting, and they're melanistic and they're small. And melanism and pygmeism often go together, but they're really small. So the males will mature at 4 inches, whereas on the mainland they'll mature. They won't mature until they maybe 15 inches, just talking in American lingo there, 15 inches. So, and they're the same species. So genetically, there's not very much difference between them at all. So this is an environmental sort of influence on the body size of these animals, which I find fascinating. And it's probably epigenetic inheritance involved in the body size, which I think is a proposition that warrants examination, and we have the tools to do that now. It's the same as the pygmy crocodiles up in Arnhem Land. So pygmy crocodiles above the falls, crocodiles below the falls get to 7 foot. Take the eggs from the pygmy crocodiles upstream, incubate them out, feed them up, and you get fat pygmies. Right? So is this an inheritance through traditional genetic mechanisms, and if so, how is that sustained when you've got the big ones and little ones in the same river? Or is it an epigenetic thing, multi-generational thing that takes a while to overcome? That's a really interesting question, I think. And it's the same with these little turtles. So is it epigenetic inheritance in the same way that famine in human beings leaves epigenetic signatures in the grandchildren after a famine, and the same happens in sheep. So that I think is really worth having a good look at now that we can look at methylomes in these animals and look for correlations in the methylation marks that go across generations. And like in humans and mammals, the methylation of our genomes is reset twice. And obliterated basically almost entirely. But in fish, that's not the case. The methylation marks aren't obliterated. So yeah, what's happening in reptiles? So in some fish, I should say, what's happening in reptiles are some of these experiential changes to the methylation patterns in the genome passed on to the offspring, and do they influence the body size? It's tractable now. Someone could do that work. Whereas when the ideas first sprang into people's heads, it was intractable. You had no way of addressing that question. So again, a young person needs to come along and have a good look at that.
What do you think about the likelihood of methylation factoring into megacephaly in turtles and development of large heads and restructuring of the cranium?
Well, I mean so methylation. There's different types of methylation, but methylation is involved in differential gene expression in cells within your body. So your heart cells are heart cells, and your liver cells are liver cells, because some genes are switched on, some genes are switched off, and methylations, either histone methylation or DNA methylations, are directly involved in that process of cellular differentiation. So when you get a developmental change like megacephaly, presumably there's some changes in the cells that are leading to that, and methylation could well be a part of driving those changes in response to, say, eating mollusks or some dietary change that occurs locally. So the animal adapts to the dietary availability phenotypically because of processes that lead to that phenotypic change, which would involve methylation. So again, you could look at that and try and work out, what genes are being switched on and switched off by methylation that are involved in megacephaly. Yeah, again, it's a tractable question now.
Yeah, it's another sort of throughout the conversation, a lot of different questions have come up. So anyone listening that's and a lot of the listeners are students pursuing turtle work, so a lot of good ideas.
Yeah. Well, what I would say is what an exciting time to be in biology, because this genomic revolution is just that. The ability to be able to read the genomes of species, the pangenome, so looking at genomics across individuals and looking at the variability, looking at the methylation of those genomes in different cell lines and how that governs development. All these things are opening up, not just for human health and in medicine, but as they forge forward with all the techniques and analyses and things, we can capitalize that in the turtle world and draw those techniques in to answer some really interesting questions in the turtle world. So, what an exciting time to be in biology if you're a young person. I mean, it's just a phenomenal future ahead in terms of the sorts of things you can do.
I think that's a really good message. There's one more thing I'm interested in touching on, and we can start to sort of wrap up. Is I'm sort of personally curious what the latest with the Michaelis georgii we spoke a bit about, but there were some issues with disease in that population. I'm curious what the latest with that is.
So the animal, so in 2015, a virus proliferated through the river at a pace that was hard to understand. It was very rapid. And the turtles succumbed to it, the adults. And so they got sick. And then they got scud as well. When turtles get sick, they tend to get secondary infections. And so they got scud, which was, all the swollen eyes and all those things that you see in captivity if the water quality deteriorates. And then they died. And so almost all of them died. So it's catastrophic, isn't it? Because they're only found in that one river. And so it's catastrophic that they would that all the adults pretty much would die. So basically, before they all succumbed to the disease, 18 animals were taken out by Western Sydney University and now Taronga Zoo, and another group of animals were taken out by another zoo. So we have an insurance colony, and they're breeding and producing offspring, and those offspring are being released back into the river to try and boost the population. So there's a number of research questions here that, again, would be a focus for study. One is those animals that survived, the adults. Did they survive because they didn't encounter the disease, or did they survive because their immune system was such that they were able to resist the disease? If that second one's the case, then you can look at the MHC genes and find out whether there's any indication as to how they managed to resist the disease. And then you can do some genetic manipulation. So you can do some breeding manipulations informed by genetics to build up that MHC complement in the animals that you're going to release. And you could, if you're allowed, you could actually manipulate that through genetic engineering to increase that diversity to release the animals into the wild. So that's a really pressing question. Did those animals that survived in the wild survive because they avoided the disease or because they were resistant? That's an important thing. The second really critical thing is whether those animals, when they're released, when they grow up to adulthood, are they all going to die? So is the species kaput, in the sense that we can do all we like to head start these little young animals from the insurance colony back into the population, but when they grow up to become adults, they succumb to this disease and die. So that's why it's so important to do that genetic work to see whether you can build in resistance to the animals you're releasing. So that's fascinating. The other fascinating aspect of this is that the Imagura got into that river and spread through it, and they don't succumb to the disease, and they're hybridizing with the George's eye in that river. So are they conveying that resistance to some of the hybrids? In which case, you've got a bit of genetic rescue going on there, or is the hybridization going to be rampant and just wipe the George's eye out through genetic swamping? There's an important question there that needs to be addressed. The jury's out on it. I don't know. We've got latisternum, for example, hybridizing with creft eye in the wild, but those two species are maintaining their identity. So maybe the same's happening with the George's eye. It's maintaining its identity, but there's a little bit of influx of genetic material from the Imagura, which may help rescue them from this terrible virus. It's a lot of work to be done there to try and see what the future for this species is. But it was interesting that the genetic work I did to look for the hybrids found one hybrid in the insurance colony, so it was taken out. So the insurance colony is now pure George's eye, disease free. So that's where genetics can assist. The other thing is that genetics shows that the genetic variability in the George's eye is very, very low. And so it's only 75 kilometres long, the range of the species in the river. And so it's probably in a bottleneck at the moment, in a genetic bottleneck, and has been for a long, long time. And it's eroded its genetic diversity. And that erosion of genetic diversity has made it like the Irish potato, particularly vulnerable to a novel virus when it does get in there. So, in terms of research, there's a lot to be done, a lot of exciting work to be done, and you can bring the new power of genetics and genomics to bear in assisting this species.
It's sort of an open question and something that's, a virus like that could wipe out the population very rapidly. So it's sort of a good thing to know conservation-wise. In terms of one more thing on that, actually, that was interesting to me is you did some sort of almost population genetic work with the Manning River turtles and the other species like Chelys, and it seemed like the differentiation in all my Chelys except for belli was fairly low. I'm interested in why that is.
So belli occupies the headwaters of the Murray-Darling River, which is our biggest river. Like it's a very large river, and it occupies the headwaters in the granitic areas. So where the substrate is granite based, and where the streams are flowing fast over a rocky bed, they occupy those headwaters, and those headwaters are isolated from each other because by the lowlands. So they occupy the headwaters and they're isolated by the lowlands, and so you've had this divergence over time in the populations in the Severn River, in the Guida River, and in the Namoi River. They've isolated over time and diverged genetically, and so the genetic divergence or genetic diversity is represented by what can be maintained in a single drainage and what the differences are across the drainages. Whereas George's eye doesn't have that. It's only in one drainage. So basically, belli's got the advantage of being split over a number of localities, each of which have maintained their own suite of genetic diversity to make the whole greater than is possible in George's eye. So that's what's happened there.
Okay.
And latisternum, of course, is a species complex. So there's more than one species there. No one's done the hard yards to separate them out, but it's going to be very genetically diverse. Certainly in the mitochondria. So mitochondria are funny. So mitochondria, like turtles, sort of Australian turtles anyway, sort of hand them out like confetti at illicit weddings. Like they sort of just hand their mitochondria across to other species. So Chelonia's expansa, for example, has lost its mitochondria altogether. So in Queensland, it's got Chelonia's cani derived mitochondria, and in Murray-Darling, it's got Chelonia's longicollis derived mitochondria, and its mitochondria has gone. So they've been exchanging little mitochondria. So with the latosternum, you've got four distinct mitochondrial clades, but the clades aren't sister to each other. So there's been some pinching of mitochondria from other species and teasing that out. So we started doing latosternum in the same way as we did the southern Emadura, but it got so complicated we just stopped and put it aside for a while until we can work out what's going on in more detail. So a lot of genetic diversity in latosternum, I suspect.
It probably is good that a lot of the early attempts were based on a protein electrophoresis as opposed to using mitochondrial genes. I can't imagine what the phylogenies would have looked like if you had used that, knowing that they're sort of
Yeah. Well, so the mitochondria is a single gene, right? So you've got gene tree issues where the mitochondrial phylogeny can be different from the phylogeny of the species that contain them. That's a bit of an issue. Whereas the allozymes, you've got 45 independent nuclear markers indirectly through the proteins. And so in a sense, the SNPs are paralleling the allozyme electrophoresis, except it's directly measuring the DNA, whereas the allozyme electrophoresis measures the proteins that arose from the DNA. So those two are more similar than the mitochondria. I think what we'll find with the mitochondrial phylogenies is that they don't always accurately reflect the phylogeny of the species that carries them. And so you get a really good, highly robust, highly supported through bootstrap mitochondrial phylogeny, but it doesn't reflect the phylogeny of the species that carry it. And the good example of that is that Chelonia's expansa. So when you do a phylogeny of the Chelonia's, you've got Chelonia's expansa sitting up with cani, and you've got other Chelonia's expansa sitting with longicollis. And that's because that little bit of sneaky shuffling of mitochondria across their species boundaries. So mitochondrial genomes, mitochondrial phylogenies have warts on them, I think.
Right. That's the genomic revolution is going to be big in terms of, as you said. So kind of going into wrapping things up, we also like to kind of focus on the adventure side of things. And we've done that and sort of weaved it through a conversation. But I'm curious, out of all the places you've worked, where in the field it's been the most interesting and your favorite place to work in the field.
Yeah, it's definitely Papua New Guinea, and I'm bitterly disappointed I didn't discover it in my 20s. So it's on Australia's border, and it's an astonishing place to work. I mean, I decided to work up in the Transfly, which is on the border of Papua New Guinea with Indonesian Papua, and it's west of the Fly River, so it's really remote. And it was so exciting. So unfortunately, the people that heralded my visit upplayed my status. And so I was a big man from, Australia, and presumably influential when I was looking for turtles. I love turtles. And so it was a bit of a miscommunication. So the villagers would spend days preparing for my arrival. And the way they work when they welcome someone is that they get in all their apparel with all their traditional gear on, and then they basically demonstrate to you that they could have killed you, but they chose not to. Right? So, and every village had a different way of scaring the living daylights out of you. So the first village I went to, they all were, in an arc, and they were playing their drums, and they had all their traditional stuff on, all the colours, and they were welcoming me into the village, and they were backing up in an arc, and I was walking forward. And then these young men are built like brick shithouses, right? They are very strong. And two of them leapt through this arc, leapt up in the air like that, and pointed bow and arrows at my chest and then released the string, but the string wasn't on the arrow, right? So they just released the string, but the arrow didn't get released. And they were literally up in the air like that. And bam. And like, I just, I was, shocked. And I was told that if I hadn't reacted in the way that I did, they would have gone to plan B. Heaven knows what that was. So that was one. And then every village I went to had a different way of doing it. And it just came as a surprise each time. One of them had a piece of bamboo that they slit and heated in a fire and pushed it down and bulge out. And they used this to kill people without damaging their skulls because they're headhunters, right? And so he leapt at me and brought this thing down within a centimetre of my forehead. And they all screamed at the same time. So like it was like they scared the wits out of me. And then they had and then the women turned up and they had two. Each of them had a basket. One, and I said, "What are these baskets for?" And they said, "Oh, that was for your head. We would put your head in that and take it back to the village." So, and then I got back to the village, and then they all were, and they did all this dancing and everything. And then I look over, and there's this guy with a head in his hand. Like it must have been from his grandfather or something, because they haven't actually killed anyone for a long time. But and when the Westerners came through, they cleared all that stuff out, right? But this one they managed to keep. So he has this shrunken head in his hand. Like, and I've got a photograph of it. Like, there it is. I didn't see it at the time. I took the photograph, and then there he is standing there with his head in his hand. And so each village in turn just tried to scare the wits out of me. And so that was just fascinating trip. To get there, we had to drive from one place to another over to Suki. We had to drive over, and they'd arranged a vehicle for me, but the vehicle, the guy had put it in for service and gone to Port Moresby. It's a nice Toyota, and they thought, oh, he won't be back for a while. We'll use his vehicle. So he stole the vehicle, gave it to us. So we're driving along in this vehicle, and next minute the police chase us down in a 4-wheel drive, and it's been a stolen vehicle. They'd pile us out, take the vehicle back. There seemed to be no consequences for anyone. And then they found another vehicle which was like a complete wreck. It had no brakes at all, and so we're screaming along cycle paths in a 4-wheel drive with no brakes at high speed and no seatbelts, nothing, with about 10 people piled in the back. Like, I don't know what OH&S would have thought of this. But that was really exciting. And then we got to the we got to the place, and then we had to pile into a canoe which is hollowed out of a tree. And there were 10 of us and a dog in this hollowed out tree with a 25 horsepower outboard going through the Suki swamps to go from village to village to collect the turtles. And that was pretty good. A big tree, 17 meters, I think it was. Massive hollowed out tree. They're adventures, right? That's and you're in the middle of nowhere, and then you end up out with people, and you've been with them for about 3 weeks, and you find that one of them's just been released from jail, for certain things, and the other one had a big split in his head where the police caught him doing something and split his head open. So you're there with these guys telling you about how their grandfathers used to eat white fellas, and they're sitting around the fire saying when the last white fella was eaten and what the circumstances were, and and all this, and then one of them will reach over and grab you by the stomach and go, greasy.
That's good.
So, I mean, that fieldwork in New Guinea is just something out of this world, and the people are so friendly and so can do. Something goes wrong, they got a way of fixing it. Doesn't matter what it is. Vehicle stolen and taken off you by the police, another vehicle turns up. Like, just can-do people and very friendly.
What's
amazing place.
What was the story with Dr. Schaefer? He mentioned that you and him had done work, but he didn't go into any detail. I'm curious about that.
I don't know. If he didn't tell you about it, it's probably best I don't. But
all right.
yeah. So he had a medical episode in Papua New Guinea, which was uniquely Papua New Guinean, and we were very remote, and the plane that took us there had a faulty engine, and so when we went to go back, it really couldn't take us back, so it took days. And then finally they put us on the plane, and I think they decided they were going to have trouble explaining how they had passengers on a plane with a faulty engine when they got back to Port Moresby, so they dropped us off at Kakori on dusk. And the story went on from there. So yeah, it's sort of interesting story, but maybe you have another interview with Brad to talk about it.
Yeah, we may have to do that. All right, well, just one more thing, and we'll sort of wrap up. This has been, adventures are really interesting. Thing I'm also curious about, you've definitely really ambitious in terms of the work that you've done and the questions you've tackled. I'm curious what the most rewarding project has been, and well, the most sort of to rephrase that, what is the most intensive project you've worked on in terms of how rigorous it was to do, and was it the most rewarding project when it was finished?
Yeah, so as a herpetologist, I guess the most rewarding project was the Sex and Dragons project. Really, it's sort of been transformational in how we think about sex determination, and it's been a passage for me because, like 15 years ago, I would have been hard pressed to explain what a gene was, right? Working through the projects which I led, funded by the ARC. I think there's been about 5 of them now. The latest one was the largest grant awarded in the year by the ARC. So we even sort of in monetary terms, we even exceeded what was given to the astronomers. So it's a really big project that involves medical institutes, the QIMR, the Garvan Institute, 2 medical institutes, all looking at the intricacies of sex in dragons. And I think really that's been really satisfying because of the people I've been able to work with who are high performing, driven individuals with a real commitment to science. So in the last grant, not this one, the last grant, I think we got 38 papers out altogether on different aspects of sex in dragons in leading journals, and this is because of the energy and dedication of the team that I was driving, and so I think that's been the most satisfying, and one of the reasons it's been the most satisfying is because I moved from what's called a mode 1 researcher, where you just work on your own. And you publish papers on your own, and you're following your own passion to, under the publish or perish mantra. And I've moved quite deliberately into the mode 2 research, where you work as part of a team, and it's partner or perish, as well as publish or perish. So I moved into that, and so it's not built into my nature to, I'm pretty introverted sort of individual. So it's not really in my nature to be able to work constructively with teams of other people. And I think that growing into that new environment and being so productive and making the discoveries we've made and seeing the excitement in the younger people when they make a discovery has all been fantastic for me. I really think it's been a crowning sort of component to my career, which I guess is going to end at some point, given I'm 70, probably got a maximum likelihood of living to 77. It's been really rewarding that those last few years. So I'd put that first.
Interesting. And sort of to end us off here, the last question we like to just inquire is if you have one piece of advice for someone looking to make turtle research or turtles part of, or just a whole career, what would that be?
I guess you make sure it's your passion, so you don't do it as a deliberate decision. It's going to be coming pretty deep from inside you that you really want to work on turtles, and they're special in terms of their life history attributes and everything that makes them so successful as a sort of lineage as fruit there for investigation, and you've got to be passionate about it. The second thing is associate yourself with people who are in well-resourced labs and who have been through it and been successful and delivered in a context that they appreciate you're in now. So you want to attach yourself with a winning lab of some sort. Don't do what I did when I was younger, and I was in a backwater really. When I was doing my PhD, and I suffered for that in some ways. I became very independent because I didn't have much assistance, but I suffered in terms of the fact that insulation and not being with the leading lights in the area. So, in recent times, perhaps not right now, but because these guys are getting older, but the Justin Congdons of the world and the people that are really pushing the foundations of understanding in the turtle world, you want to identify them and associate yourself with them as a student or as a postdoctoral fellow. And then the other thing is just get skilled up. The world's changing. It's changing really rapidly in terms of skill sets. Get skilled up. So be the best at least some elements of the skills that you need to do your work, so that you've got something to come to the table with in terms of collaboration with other people. So if that's modeling or programming or life history analysis or some aspect or genetics, be good at something so that when you approach other people for collaborations, you've got something to bring to the table. So that'd be the three things, make sure you got a passion, make sure you link up with people who are leading lights in your field that you can launch off their shoulders, and then the last thing is make sure that you keep up with the latest in the technologies that are coming to bear in areas like morphology, where you got 3D scanning and all that. Make sure you're up to breast with at least one of those, so that you've got something to bring to the table in collaborations. Yeah,
I think that's a really holistic overview of things, and we'll definitely all take that. I think should definitely do the same. I think that it's really great advice. So it sort of is it, unless someone else has anything that you want to say. We will at the end do a little round of turtle trivia. We've done this 44 times with about 50 guests now. Here it comes. I try to mention it to everyone beforehand, but I forgot, and I've forgotten for the past 20 or so guests. But I don't know if you've got a obscure turtle trivia question that you want to ask us, or we can throw one your way, however you want to do it.
Trivia question. I mean, the one we get asked in Australia the most is, what's the difference between a turtle and a tortoise? Because in Australia we used to call them all tortoises, and then we've gradually moved to the international norms of calling anything that swims in freshwater a turtle. And so that's the question I get asked the most, and my answer is it's all in the spelling.
Yeah.
I don't know if that's trivial enough for you, but
in terms of the fact that just all tortoises are turtles, not all turtles are tortoises, that sort of, or is there something more to that?
No, I think so. Basically, we don't have tortoises in the sense of desert tortoises. And so we made a distinction between marine turtles and freshwater tortoises. And they still do. So in Western Australia, they still call it the Western Swamp Tortoise, even though it's a turtle. So it still persists. And so in the public mind, that's the question that always comes up. Even if I'm talking at a primary school or something, the hand will go up and out will come that question. I don't know if there's anything else. I mean, I get that question. When I'm giving a talk, I've got the turtle in my hand, and someone will put the hand up and say, you're not supposed to hold turtles upside down because they don't have a diaphragm, they can't breathe. Well, I don't know if that's true or not. I don't know where it comes from because they've got a solid shell, so it's not as if the viscera might rearrange, but and also a turtle can hold its breath for 2 hours. So that's a bit of trivia, and I don't know whether it's true or not. So it needs to be in a trivia quiz so that we can make a decision as to whether it's true or not.
Well, that's good. I mean, that's some good questions. Stuff to think about too. It's sort of open-ended questions in some ways. So,
So what's your trivia question for me?
Oh well, we can throw one your way. It's again on the spot. I don't know, Jack or Ken. Do you guys have one on the top of your mind here?
Trying to construct something, but I don't know if we'll be quick enough.
I'm not letting you off the hook. You put me on the hook. Now you're on the hook.
We even said we would do it. Okay, I've got one. I've got one. This is one that's appropriate for Jack and I and a few other guys were out in the field a few days ago working with graptomes, map turtles. In terms of this is a kind of a Jack question, but I'm asking it. Which map turtle has the widest and largest head of all 14 species?
I've got no idea.
It's the one we were working with, Barber's map turtle. They're something else. They've got just insanely large heads for crushing mollusks. The females do, at least. Males, you could probably make an argument. I bet they're also kind of the largest in terms of males, but
they're like some of the female Emidura, like that get super megacephalic, like really,
right, okay.
really. But their body mass is like 25 times that of the males. So yeah.
Yeah, right. Yeah, sorry, not that familiar. I've only been to the US a couple of times, so I'm not really familiar with that one. So you got me on that one. I think we have to have an Aussie trivia competition that's like not, and you can have your own US trivia competition.
That's fair. All right.
Yeah.
Jack or Ken, you guys have a Australian turtle related question? That's a tougher one because
What do you think?
I've got one. I've got one related to Australian turtles that came up. We recently had Scott Thompson and Gerald Kukling on to talk about the Western swamp tortoise turtle, and so they at one point they fenced in the habitat for that species, and there was a problem with the fencing in. They thought it would help, but it didn't help. What did the fencing do that was not good for the turtles?
Yeah, so there's such a thing called the fence effect. I don't know if you've seen Krebs's work on the fence effect, where they put a fence around a particular area, and the voles went through the roof in population numbers and ate themselves out of house and home. Because dispersal is a really important component of population regulation in some species, and in bandicoots it appears that's probably similar. And so when they put the fence around, the bandicoot numbers built up because they weren't dispersing, and then they started eating the turtle eggs. So basically they had that problem of fence effect, driving up the population artificially of the bandicoots, and that having an impact on the turtles, as I understand it. But maybe I'm wrong.
Nope, that was right. And I was not aware that the fence effect was something that was well established. So learned in the process of asking the question.
Yeah, that's all right. So they need to put little trapdoors that let the bandicoots out, but not back in, if they want to alleviate the fence effect. We have the same thing in our sort of reserves in Canberra. We have a fenced Mulligan's Flat, and there's a fence effect there that requires management of the population numbers inside, and they can manage the grey kangaroo numbers, which would naturally go up as a result of that fence effect. But they can't manage the redneck wallabies because they're a native species that's not able to be managed in the same way legally, and so they've gone through the roof, eating the place out of house and home, and that dramatically affects all the other species in there, including the long-necked turtles. So that fence effect is something that needs to be recognised and managed very actively.
Interesting. That's an issue across Australia, not just with the swamp turtles. So it's good to know. All right. Well, we could throw one more, or we can wrap up if Jack or Ken, if you guys have something. But we've been going for a while, but it's been really an honor. Dr. Georges, thanks for sitting with us and telling us your work, and it's really awesome.
No, it's been really interesting for me as well, and great to see you guys are so enthusiastic about getting this out to a broader audience, and the amount of work you put into the questions and the depth of your knowledge goes beyond what I'm normally confronted with when I'm talking, say, to journalists. So, I just commend you on that. It's been really a pleasure talking to you guys. So, hopefully, if you come out to Australia, you can catch up. We can catch up. So, let us know if you're ever coming out this way.
Go ahead, Jack.
No, not saying anything.
Oh, I thought, okay. Yeah. Well, we'll definitely take you up on that. And it's on our list of turtle life lists and just going exploring. So thanks again. And we'll sign off for now. So for everyone,
Okay.
this is CheloniaCast, episode 44. We'll see you next time.